Thanks everyone for coming. Welcome to the RIEL Seminar today. We've got Carol Palmer and Natalie Robson talking about marine megafauna research. I should also acknowledge we're on Larrakia country, so it's acknowledge the traditional owners and acknowledge their elders. So the research we're talking about today is really cool work on marine megafauna that's been going for 2021. The ARC linkage project started, wasn't it, Carol?
Yeah.
And it's been a big project that's brought together CDU, a whole lot of different ranger groups, Taronga, James Cook Uni, Macquarie Uni, and plenty of others who can be offended by me forgetting them. And so Carol's been leading that project in the field and here at CDU, and Natalie's been a PhD student who started on the project, has been doing fantastic work on green turtles. It's been a really good, as well as the science, it's been a really good partnership. It's had a lot of ranger groups involved with it. And one of the really important things about this project is, as well as the funding that we got through CDU, Larrakia Rangers sort of in parallel got a lot of funding to support the ranger-led work from a ranger grant through NTG Infex that was funded there. So it's really, really good working with them. And recently, Carol and Nat have also secured some Commonwealth Marine Parks funding to work up in the Arafura Marine Park with the Gunnery Rangers up on Croker Island. So it's like I said, been a good research project, but probably almost more important than that, really good network building project and getting people together working on marine science and marine megafauna together, and just building a really supportive group of rangers and researchers to work around the Northern Territory. And for someone who's a complete non-expert in marine stuff, from my perspective, it's a great opportunity to actually, Nat might roll her eyes. I've been on bugger all field work with her, but actually down here and do a little bit of film. Anyway, that's probably enough for me. I'll pass over to Carol. You're starting off, aren't you? And Nat was
We're sort of starting together.
Then follow up with it.
But if I could just say, Sam is a marine expert. Don't listen to what he says. Okay. Well, thanks everyone for coming. So we'll just do a quick kind of run over of some of the cetacean work, the ranger exchange, because we've worked with 8 rangers, and then we'll be handing, Nat and I will do this bit together, but then we'll be handing over to Nat for her fantastic analysis with the green turtles foraging in the top end, and that's the first time this work has ever been undertaken in the NT. So it's really nice that we're moving forward. It's a bit too slow, but we are actually moving forward. So how do I do this? Just, oh, that one. So this is, yep, so which one, that one? One. So yeah, that's just at the very start at Kakadu, where we tagged 2 male green turtles for the first time, but now I'll talk a little bit about what they did and where they went. And this is really just to highlight, look, NTC country, we really are very important for dugongs, turtles, whales, and dolphins. And yeah, most people don't appreciate how important we are. So hopefully with really good communication and stuff, we can get that out to the rest of Australia, if not the globe. Oh, sorry. And this is just to highlight the areas that we've worked. It's a bit of a weird map, sorry. But this is the ranger groups that we've worked with, and where those are circled, that's where we've worked. And this is really just to highlight, this is the ranger exchange with us. They're sea people, they're extremely good at radio, extremely good eyesight, and we recorded quite a lot of things. And that's with the Kakadu mob, Coburg, Croker, which is the Gunnery Rangers, Wessels, Fog Bay. And this is Larrakia, Tiwi, Marabooka, Kendi. And yeah, you can see it's really great work that we've all done together. And we've run 2 workshops. This is in February 23. And this was at the CDU Waterfront, which was actually really lovely, mainly with the ranger groups that we've been working with. This is this year, February 24, and this does include WWF. And what else to do with WWF?
Joe came from Taronga.
Yeah, Taronga Zoo.
The Environment Centre.
Oh, the Environment Centre. Yeah, so it was kind of really great that we could just talk about a whole range of things. And this is just to show you, so this is just a very quick video of the rotation. Do I just pick?
I think you can just press the next.
I might get you to do that. And this is really just to show you, and if you've got any questions, because most people don't appreciate the rodeo, safety is our number one. We've had no issues with crocodiles because we are so safe. And this is with all the ranger groups. That's with the Canby Rangers, Larrakia and us, and catching the turtles, putting the sat tags on, taking DNA measurements. This is with the Coburg Rangers, and you'll see Nat jump over the board and catch a turtle in a second. There she goes. And wait till you see this huge green turtle that she catches. It's lost its front flipper. And this is with the Coburg Rangers. And people, we just love this work. And this is the first time this is being done where we're getting this information and sat tagging them, DNA. This is with the Gulliga Rangers, the Croker Island mob. This is Nat with her. I'm filming underwater. You'll see this female release in a second. But everyone just loved working together as a team. And we've got some underwater footage of some bottlenose dolphins. It's pretty terrible, but they come over and check us out as much as we check them out. This is at Coburg. And these are just the bottlenose dolphins. We have a number of species that always come into the bow of the boat where we can get DNA samples using a pole and images and record what they're doing. Just that. And then, and this is really just to say, you know, we have all these species across the top end, which most people don't realise. And certainly humpback whales are really important from a traditional point of view, as are a number of species, as are the turtles, dolphins, whales, and manta rays. A whole range of species from a traditional point of view are 100% important. And these are humpback whale figures that we've taken in the top end. Oh, sorry. And this is just a highlight. So this is just quickly with the reef manta rays. We've done DNA sampling. We've sent it to our colleagues in Queensland. It does look like our coastal reef mantas here are a unique population, but we need to do more work because they're critically, well, they're certainly endangered. We've got to do more work on them. We haven't got any funding yet, but we'll keep doing DNA samples. These are the species that we've recorded, and a number have never been recorded before. I won't go into it because we'll hand over to Nat in a second, but you can see where we've recorded all our dolphins and whales. With the false killer whales, this is some satellite tagging done years ago, but with the DNA sampling, working with our colleagues in America and the Kimberley by WA, we identified that we have a unique coastal population of false killer whales. Our New Zealand colleagues have just done their analysis of all the Pacific Island false killer whales, which are offshore species, and they've confirmed that our species, this was released like a month ago, that yes, we have a unique coastal population of false killer whales as they do in Hawaii. So we've got to actually update their conservation status because they are in low numbers and they're amazing animals and they live for as long as we do. And finally, I've just put in this effect, which I never really knew much about, the biologically important areas under the Commonwealth. So we've flagged the snub-fin dolphins based on the foraging, their behaviour, and that's the area that I flagged, which we should identify as a biologically important area. The snub-fin dolphins have declined significantly in the NT, and the humpback dolphins, the humpback dolphins. So it's the same area, it's the two species, and we're updating hopefully their conservation status. We've been trying to do it for many years, but hopefully we've got it. And finally, I don't know, many people don't realise here are the sperm whales, where have they stranded and died? Tiwi Islands, and most people don't realise that. And yeah, we've got a lot of work to do on a whole range of things with our rangers, traditional owners, Charles Darwin University. We've got a lot of work to do. And okay, it's over to Nat, who's done fantastic work and analysis, and this is the first time this has been done in the NT.
Thank you. Oh, sorry. I've been losing my voice a little bit, so bear with me. Hopefully it'll go during the presentation. So I'm just going to do a little bit of a rundown on my PhD and the results that we've got so far from it. So the overall aim, I guess, of my PhD has been to inform ecosystem-based management of turtles. So looking at all the different influences and how we can conserve them better. So my chapters are inferring the migration routes through genetics, so whole mitochondrial DNA analysis of two of the main foraging areas. So turtles always go back to where they were born to nest and to mate. So by looking at the genetics of the foraging turtles, we can get an idea of those migration routes that they'll do every 5 years. Chapter 2 is defining the habitats used by foraging green turtles in the top end. Chapter 3 is the fine-scale habitat use and diet of green turtles foraging in the NT. And then lastly, integrating expert knowledge into a spatially explicit risk assessment of green turtles. So, as Carol mentioned, we've got ranger groups that we're working with across the Northern Territory. For most of my chapters are focused just on 2 sites. So Treepang Bay up at Coburg Peninsula and Garrick Island Marine Park, and then Field Island in Kakadu. So you can kind of tell just by looking at this why I've chosen those 2 sites. We've been able to catch a lot more turtles there. So I've got 50 green turtle genetic samples from Treepang Bay and 40 from Field Island. So you can see on the right there all the different genetic stocks that we currently know in Australia that's done using a control region of the mitochondrial DNA. At the moment, there's a group in Queensland and other areas that are looking at the whole mitochondrial DNA to try and split up those genetic groups a little bit more. For example, in WA, between the Pilbara and the Kimberley. I've done that before with SNPs and found there was a slight difference, but not significant. So we're trying to look at whole mitochondrial DNA. The reason we use mitochondrial as well is because it's maternally inherited. And we're looking at the nesting beaches because they always go back to that same place. So that gives us a really good idea. Look, this is where we've caught those turtles. Give you an idea on the reef of where we've been able to catch them, which leads into my next chapter of why they're using certain areas. So I've been trying to map the habitats of these 2 sites. So I've used an underwater video system, which is quite a simple system that I made myself. And the reason I use that is because it's quite easy to get the materials for, it's easy to use, it's quite lightweight, and it's quite small to take on our little boats out in these remote areas. A couple of the ranger groups have now made their own versions as well. So it's really easy for them to use, and it's easy for them to then continue monitoring and doing longer term surveys, which a lot of groups have said that they want to do. So these are my ground truth transects at each site. And I've gone through and analysed all of those images. It's a little bit hard to see on here, but these are my 2 sites. So I've gone and done a cluster analysis and grouped the transects based on their similarity and how similar they are. And then you can kind of see on here, so we've got these ones at the top here, the Treepang Bay sites that are more driven by mud and silt. We've got the corals down here, seagrass and coarse sand at the top. So I've kind of used this analysis to come up with biotope classifications for our habitats, and then used remote sensing to map the whole area. So this is my Treepang Bay map. Just to give you an example of what this means. Got this one here, which is our deeper subtidal mixed coral reef and seasonal seagrass. So that's a lot of big soft corals and sponges. And then during September, October, November, more like November, December, we've started noticing dense seagrass there, which we've only done a little bit of surveys outside that to show that it wasn't there all year round. So a lot more work kind of needs to be done on that. This is our intertidal seagrass. Coral reefs. So the percentages there is just how percentage of the mapping area made up by that habitat type. This is the intertidal algal reef. Field Island. So this site was obviously a lot emptier. There's a lot more freshwater input within the Van Diemen's Gulf, which meant that we had to be really careful about what tides we do the transects at, so you can actually see anything. But it still worked out pretty well. So again, we've got that seagrass. This is the mud silt and seagrass. You know, we've got intertidal coarse sand and seagrass. Coral reefs. So the species of coral were quite different out of Field Island compared to Coburg. So we're getting a lot more of these big flake corals compared to Coburg, which is a little bit more branching, probably because of the big tides that they've got there. And then algae, mixed coral and algae. So just going to show another video. So the next stage of the PhD is looking at how the turtles are actually using this habitat. So first we've got to catch them. This girl had seagrass in her mouth, which helps us identify what the diet are eating. She wasn't pleased that I interrupted her lunch. So they're pretty heavy, sometimes to get up onto the boat. Get them up there, would measure them, get the length and the width, which I'll talk about in a minute. And then we attach the satellite tag. So to attach the tags to the greens, we use, so I'm just sandpapering the shell to get rid of any ridges and get rid of algae that's growing on the shell. And then we use a mix of fiberglass and epoxy glue to stick that on. And then I'm doing these cloacal swabs for eDNA and diet analysis, which has only been done once before in by some Portuguese scientists, so it's quite a new technique for the turtles. So I've analysed the satellite tracking data of these turtles and looked at their core foraging activity space, so their 50% kernel utilisation density. So for Coburg, the areas range from between 4 to 14 square kilometres. There's not very much overlap between the turtles that we've tagged. As you can see, where they're foraging is quite spread out. They're moving around quite a lot. Overlay that onto the habitat map. So I've only just started this analysis. This is just the first step. You can see there's been quite a lot of time on the deepest subtidal mud. That's probably because of the tides. They tend to move off the reef at the tides and then come back on to eat afterwards. And then the intertidal seagrass, they're using quite a lot. You'll notice that one turtle is quite different, 25 to 665. This one spent most of its time in that deeper mixed coral and seasonal seagrass habitat, and that tag actually only lasted during that time when that seasonal seagrass would be there. So it's a little bit of a skewed result, and if the tag had lasted a bit longer, the results might have been a little bit different. Field Island. So as you can see, they're using a much smaller foraging area and they're not moving around as much. They're kind of sitting in the same spot compared to Coburg. It's also, the reef is also a lot smaller, so that makes sense. And the overlap, maximum overlap was about 67% between those turtles. So unlike Coburg, the turtles weren't really using that intertidal sand and seagrass habitat very much. They were spending most of their time on the mixed coral and algal reef habitat. And that makes sense with what the rangers have told us, where they seem to think when they're eating turtles out there that they're eating algae, they're eating red algae, compared to Coburg where we catch them with seagrass in their mouth. So we think that the turtles are eating different, or preferencing different habitat and species. And that impacts their growth rate quite a lot. So certain species of seagrass have higher fibre and mean to get much higher growth rates in turtles compared to algae. And you can see here the Coburg turtles are much bigger than Field Island. So it could be because of diet, or it could be that certain age classes are preferencing different habitats. There's still a lot of questions to be answered around that. So for the EDNA, going out to Capitol again to get some more samples to try and bulk that up before we finish the analysis of that one. And lastly, my human impacts, my risk assessment chapter. So there's 3 stages of this risk assessment, which is identifying the hazards and looking at the cumulative pressure, mapping the cumulative pressure. The exposure assessment, so where turtles are actually exposed to those hazards, and then vulnerability, so what degree are the turtles actually being affected by those hazards. So for the hazard assessment, I've got data layers, I've sorted them into different categories, and then the hazard and then which behaviour is impacted. So I've done separate maps for foraging and migrating behaviour, which looks like this. For foraging turtles, obviously, probably unsurprisingly, Darwin has the most number of cumulative threats. And this is the migrating pressures. So for turtle movements, I've included not only our satellite tracking data, but any data from any project where the turtles have passed through the Northern Territory. So it's about 20 years worth of tracking data. So that's from Kendali, Impex, Woodside, a bunch of other AIMS have all contributed data, and that can use that. So
Except these ones are the ones we've got are the foraging turtles, not the
Yeah,
nesting turtles.
yeah.
That's the
so it's a
difference.
mix of nesting and
Yeah.
foraging. So I've gone through and looked at time spent per grid cell on like a 2 kilometre square grid for all of those turtles and summed it to look at which areas are most used by turtles and then more likely to interact with the threat based on their behaviour. So their behaviour was defined by their speed and turning angles. So I then split their tracks by that and analysed it by time. So this is their occupancy index for foraging. So I'll just go into that in a bit more detail. This is the Darwin region. So we get them around Channel Island and in Bino Harbour, around Bear Sand Island, and then out at Middle Reef as well. The Tiwis, scattered all around the Tiwis. Field Island, and then the Coburg Peninsula region. So the top here, that's Truco Bay, that's my site. They're foraging in other areas of it as well. Migrating. So migrating, they obviously spend a lot less time in a cell as they're passing through because they're moving quite quickly. The impact tends to be a little bit less. I've just changed the scale of this so you can actually see the tracks because otherwise you can't actually see them. So then I multiply the occupancy index by the cumulative pressures to get this exposure intensity, which is which areas are most affected. So as you can see, the value for the migrating is a lot lower. So that's still out of between 0 and 1, but the values of each cell are quite low because the turtles are less likely to actually interact with one of those hazards. So lastly, I've got the vulnerability assessment. So this kind of gives more of a real world impact and fills in the gaps in the data because there are a lot of gaps in how realistic this data is in those remote areas. So the way I'm doing this is expert opinion surveys with the ranger groups that I work with, being the people that are out there all the time dealing with these issues. So I've gone out so far, I've interviewed Larrakia and Kemby. I've got plans to interview the other 7 groups that we work with. So I've come up with a vulnerability score. So each ranger has been asked to assess each hazard, rank the impact that it's having that they think it's having to turtles, how often it's impacting them, and how confident they are on their knowledge of that hazard. So the vulnerability score is those 3 multiplied together. So for Darwin and Bino, Larrakia and Kemby, this is kind of how it's looking. So I've ranked it as low, medium, high, and extreme. So as you can see, agriculture, mining, sewage runoff is coming up as a big one for both ranger groups. The big spike for Kemby, which I didn't expect, and every single ranger that I spoke to mentioned the lithium mine out of Cox Peninsula and that they were really sure that that was impacting Bino Harbour every wet season. So that I didn't expect that. In Darwin, it was more the PFAS and sewage runoff that they were concerned about. Sea level rise and erosion was obviously a big one, and plastics as well. And then recreational fishing and vessel strikes came up pretty consistently as a concern across all groups. So cumulatively, even though there's a few spikes in Bino Harbour, Darwin's still got the biggest cumulative threats, which again, is not particularly surprising. So the next stage, when I finish all these vulnerability assessments, is to stack them again onto the hazards and the exposure, and then get this final risk map of where the turtles are most at risk in the Northern Territory. And that's it.
Well done. And this bit's just to show you what we've identified over the last chunk of years, what working with traditional owners, ranger groups, all our other colleagues and things like that, we're seeing sea level rise, sea depth rise. We think nesting temp erosion. We're worried a bit about seabirds because there's not many people working on seabirds here across the top end. Bycatch strandings, on and on it goes. And you can see these mangroves, you can see that it's kicking in. And then the next one is plastics and ghost nets, and you would all understand and have seen so much about this, but it really is a huge deal. And it's so tough for so many people in these remote locations to collect the ghost nets, collect the plastics. We're not doing much water quality testing at these other places, which we really do need to step forward just for people's, not only just, our marine health, but our people's health too. And that's it. And also from all the traditional owners I've worked with over many years, they all say it is the dolphins, the turtles, the whales that sink back to your country. So thank you. See you.
Any questions? Yeah.
Thank you. That was great. Thanks very much. Really interesting. I was wondering about the dietary differences that you observed. And you gave several possibilities. You said there's still others, but I wondered if you have a best guess hypothesis that you're testing. What's your
one of the options is that we've seen a lot of dugongs at Field Island, so they could be foraging in that seagrass area, which means there's a bit of competition with the turtles. So the turtles are eating the algae is one option. So I need to map we've got GPS locations of all the dugongs we've spotted throughout the project. So I'm going to map those and just have a look with the turtle foraging as well, and just see if that's an impact. I'll know more once we get our diet analysis back, our EDNA diet analysis. And I'm also just going to have a look at do a bit of a deeper dive into the literature. There hasn't been a lot on diet analysis so much in Australia, mostly stable isotopes and around Ingaloo they've done a lot. There's been a fair bit in the US, so I'm just going to have a look at that as well, see what they find. But it seems to be fairly site-specific at times from what I'm finding. But yeah, just keep digging.
Yeah, I mean, you mentioned the age thing, and I was wondering whether there might even be a life history.
Yeah, potentially. I think we'd need to look at a few more sites. I've definitely noticed areas like Darwin, which seems to only have juvenile breeds, and then adult hawksbills. Same with Bino Harbour, seems to be mostly juvenile breeds, whereas Field Island and Coburg definitely have the bigger turtles. But as you saw, I didn't even realise. I kind of suspected the Coburg ones felt heavier, but until I mapped it out properly and looked and saw that there was actually quite a big significant difference between the sizes of the two sites. So, yeah.
Thank you.
Thanks for that. It's really great background. I was also interested in the foraging habitat and the movements, and I was thinking in relation to the vulnerability assessment that you were doing, or different groups were considering. Was part of that impacts of climate change on the food resources?
Yeah.
Yeah.
Yep, so that's why the foraging and the migrating risks were different. So things like potential smothering of habitat from dredging and things like that I've included in there, as well as like sea surface temperatures on impacting the habitats, trawling scars and things like that through the habitat was in my ranger surveys, if I've seen any of that. So yeah, I'm trying to include that in there.
And just another thing worth mentioning, but I don't know if anyone's looked at, that I was really interested that you brought up is differences in the quality of food. So the preferences and whether or not, it'd be really interesting to know the nutrient composition of these different food resources and whether or not, we have some data with fish that show you that, before breeding and things like that, the preferential selection for particular food resources that have, high fatty acid profiles or things like that, and whether or not when you finish your eDNA analysis, of course, are they preferentially selecting food that has different qualities, and as you say, for different reasons, juveniles, development, or breeding females?
Yeah, definitely. I need to do again a deeper review now that I've kind of started looking into this a little bit more and seeing my results. I know my old honours supervisor, Scott Whiting, did his PhD up in Darwin and never published it. But his was on the foraging ecology of turtles up here, and he looked into that quite a lot. So he's the one who told me about the fibre and the seagrass versus algae and why they preference certain ones.
Yeah.
But that's not published, so that's just him. So yeah, I need to do a bit more looking into it.
The sort of threat scores, are they directly comparable to similar metrics elsewhere? Like you could, sort of clearly Darwin's got the highest threat ratings for some, around the Northern Territory, but there's always this perception that, no matter what, nothing's that bad in the territory. But can we directly compare that using the same metrics to other areas, or is it?
I haven't really looked into it, but I doubt it. There hasn't been a lot done using that method, particularly.
But you could if you had the data, like the same scaling, whatever would apply.
Most of the expert opinions that have been done with other scientists and more of a global scale. So yeah, it hasn't been done very much using Indigenous people or ranger groups to be the experts. So there's not really a lot to compare. It's relative within the Northern Territory and the people that I'm interviewing. And it also depends on the number of rangers there are as well. Like Darwin had 15 and Kempy's 4. So the results are probably skewed a little bit by that as well.
It's interesting though that so much of the movement data converges on going around the corner in the sort of northwestern top end though as well. So that potentially quite impactful threats in that location.
That little corridor between Darwin and the Tiwis going up seems to be a big one for foraging in that area as well as migrating through that area because there's so much traffic going through and all the industry.
Can I just ask a question? I always considered, and I could be completely wrong here, that's why I'm asking you, seagrasses, in my experience, they dominated in places like WA, South West WA, which I was used to seeing when I was talking. And I'd always thought of them as more temperate species, and then you get into the coral and the algae when you come further north into the tropics. So is seagrass as dominant here as it is further south?
I've seen some sites that are quite dense in seagrass in different species, and I know there are some seagrass mapping projects going on at the moment in the NT. But yeah, I was surprised at first. I thought it was just quite patchy bits of seagrass here and there. Some of the ranger groups have mentioned they've noticed a loss in seagrass in some areas. So I'm yet to visit that. But yeah, definitely around Coburg, I've seen quite dense meadows of seagrass. At the deeper sites as well as quite shallow at the right time. You've got to get the time of year right a little bit when you're looking at it. But also out in like Raffles Bay over at Coburg had the species now. But quite long, really dense meadows of seagrass that we drove over on the boat and we could just see it. So we definitely have it here. I just don't think anyone's done much work.
And it's a bit tricky for the rangers to do that work. They really do need people like us to help pull the information together. And the Elcho Island, the Markapal Rangers, they're saying that they've got a real hotspot of seagrass. We haven't had a chance to go over that with them. Hopefully at some point we do, but again, it's just because these remote work, we haven't done a lot, and hopefully all this stuff flags that we yeah, can kick in other PhD or masters or whatever students, but other scientists and rangers and traditional owners to kind of pull the two bits of info together. But yeah, I think, and because, yeah, we're a bit unusual compared to the east and the west coast up here, the top end, our tides, our shallow waters, you know, we're very different, a little bit like the Kimberley in part. But yeah, it's very interesting.
Yeah, we've even seen quite dense seagrass out at Bino Harbour, the top of
Yeah.
Indian Island. We sat there and saw about 10 dugongs in 5 minutes, so we dropped the camera down just to have a quick look. Didn't
And that was
do anything
August
properly,
last
but
year.
yeah, there's quite a lot around there too. A lot more to be done looking into that sort of thing. Even the seasonality and kind of narrowing that down up here probably needs to be done. It seems to be similar to the east and west, but no, I don't think we've actually done that properly.
Yeah, it's great. That's it.
Thank you.
Thank you.